This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 249, recorded on September 6th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today is no one. It's just me.
That's right. I'm a hologram. I'm sitting over here, but I'm not really here at all.
There he is, right here in the studio, Dickson, Despommier.
I sent this hologram all the way from Australia because that's where I was.
It's breaking up, Dickson.
I'm sorry about that.
How are you, Dickson?
I'm fine, Vincent.
Welcome back.
Thank you so much.
Good to see you 2 weeks in a row. I think that's a record.
It is. It's a bit of a record. That's true. It's not a broken record, however.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody. How you doing?
Tuna fish.
Tuna fish, yes.
That's right. In B flat.
For the listeners out there, that's what I had for lunch.
Right.
Tuna fish.
That was the sound check.
And importantly, let me get this here straight. It is 93 degrees Fahrenheit here in sunny Florida.
Goodness.
That's 33.9 Celsius.
Wow.
About a half cloudy sky, but, mostly sunny, actually.
Humidity?
It looks like it could be some thunderstorms this afternoon. I don't know.
Any humidity?
Well, let me see here. I don't have those data with me.
No. Well,
Listen,
sorry.
You hear him? He said those data. He's good.
Yeah, not bad.
I practice.
Here, it's chilly. It's 21 C.
It is.
This morning was 13 when I drove in.
It's perfect.
Blue sky, not a cloud in the sky.
Nope.
33% humidity.
Vince, you misspeak.
What?
There are clouds in the sky.
There are?
Yeah, take a look at the horizon.
I don't include the horizon.
I can see clouds.
I couldn't see them from my vantage point.
Well, I know that.
And the wind is negligible, 5 kilometers per hour out of the north.
And it's low humidity. I don't know what it is, but it's...
Here we go, 46% humidity with a dew point 69 degrees.
Right.
Also joining us from western Massachusetts, Alan Dove.
Good to be here.
You must have weather like me, right? It's kind of chilly.
It's pretty similar. It's 63.9 Fahrenheit.
All right.
Winds are light and variable. We've got a few clouds at 5,500 feet, and then some broken, mostly cloudy layer at 25,000.
Right. Where'd you get all that detail?
I am looking at the Weather Underground site, which has all this information.
Wow.
Barometric pressure 30.19 inches.
We have a pick later on with some weather sites.
I have a good pick this week, too.
If we get to it.
I like the Weather Underground site because it's got local weather stations that people have just set up on their houses, and you can look at data from those, too.
That, of course, is a takeoff on the other weatherman.
Yes.
Weather Underground?
Yeah.
Wasn't that someone's pick one week?
I don't know.
I think it must have been.
Yes. I think we have another bunch of these coming up.
Yeah. All right, let's get going here. Follow up. Suzanne writes about the Vax stamp. This is a cool idea. Instead of a heart, though, it might be neat to see them leave a stamp that would last a little while with the vaccine's initials. My kids' doctors, or the nurse or PA actually, would write them on the Band-Aids the first few times when there were multiple shots, so we'd have an idea which was which in case there was any swelling or local allergic reactions in the following few hours. But even though my kids were infants at the time, they were pretty good at getting rid of the Band-Aids.
Right.
That's a good idea.
Yeah.
You put the vaccine on the heart.
Absolutely.
Maybe you do a permanent tattoo, right?
No.
I don't think so.
And
Well, it depends on the vaccine, I guess. You do a permanent tattoo with the smallpox vaccine.
You do.
That's true.
We don't give that anymore, though.
No.
unless you're in the military,
Well,
which you're not, I know.
No.
Because you're a peacenik, right?
You betcha.
Or in my lab. You can come work in my lab. You'll get a smallpox vaccination.
But you better hurry up, Dickson.
Yes. Otherwise, I'll forget what a lab looks like.
No, it's closing in a couple of years.
Oh, yeah.
The next one is from Anne, who writes, Hi, TWiV gang. I was bummed to miss your session at Harvard last month. Episode 244, Experiments kept me too busy. I just wanted to request that you update the link for our student group, Science in the News. Jeff's pick of the week, we still have some confusing problems with redirecting to the old inactive website. So please post this link to our new and active website. All right, I will do that. I was supposed to do it by today, but I didn't. So now that I read it, I will do it. And finally, Stefan writes, Dear TWiV team, I was just listening to TWiV 247, and I really like the idea of a TWiV index and summary for each episode. I extensively integrate TWiV episodes in my classes, and I find it more and more difficult to remember in which episode you were talking about certain issues. Funny titles like Today's Weather and Virology work in my short-term memory, but don't last too long. Since this is quite a task for one person, how about putting this out as a project for your TWiV listeners? With 10,000 followers a week, you probably can get this done between episodes. Maybe a public Dropbox folder with template file would work, or a TWiV wiki. I'm sure there are tons of other possibilities. As Kathy suggested, this can also include keywords, long-forgotten ideas, as well as show topics and guests.
Wow.
I think someone suggested a wiki last time, too. Mm-hmm. It's a good idea where people can put stuff. Great, keep up the great work, and special greetings to Kathy. Hearing her on TWiV reminds me of our SWIG meetings and makes me feel home. Go Blue! Best from Lübeck, Germany, the capital of marzipan.
Oh.
I didn't know that.
Germany.
I didn't know that.
I didn't know that either.
Then there's a Lübeck, Maine, too, but did you know that?
No.
It's probably their, I wouldn't call it sister city because that's sexist to say that, but there are cities that are in common, like Perth Amboy, New Jersey.
Yeah, really? What's that?
And there's a Perth Amboy in France.
Oh.
New Jersey was first, right?
Probably.
Stefan is Stefan Taub, who is now an assistant professor at the University of Lübeck, which is in Germany. And he used to be at the University of Michigan. He was a postdoc with Christiana Vobus.
Right.
Who was...
Is he the one who made you the 3D printed polio?
He was indeed.
All right.
Which Dickson has managed to shatter.
Oh, I did not.
Did you break it, Dickson?
No, I just bumped off a few receptor sites.
Which I have since put back on with crazy glue. Yes, he did that. He was my host when I visited, and you visited at the same time, right?
That was a good trip. That was a great trip.
3D printer.
And in fact, the paper we discussed with Christiana on, infection of mice with norovirus, with human noro, that was his work.
Okay.
So I hope everything is going well. That's a good idea. All right, moving on. Isn't there a show where they say that?
From Norwalk, Ohio, by the way.
He's the one who told you were wrong. It wasn't Norwalk, Connecticut.
I could never forget how humiliated I felt after that submission of my ignorance. No,
that wasn't the last time.
No, of course not, but that was the most egregious.
We all make mistakes, Dickson.
We do, and that was a mythology that I had perpetuated for many years in my own group.
I got an email today about a TWiP thing that you said, but this is not TWiP, so I won't read it.
Uh-oh.
Next time we record TWiP, we can embarrass you on every...
We'll name that one Me and My Big Mouth.
Dickson, you could be embarrassed on every podcast.
Every day, and every moment of every day.
At least you laugh about it.
You try.
Our paper today, so we have been intending to do all email for several weeks, but papers come out that I can't resist. So we'll do one paper. And it came out. They
sent us a paper we couldn't resist.
We couldn't refuse the paper, is what you meant.
So I have to tell you, Carl Zimmer sent me this paper last Friday for comment under embargo.
I know which one this is.
And then it was published on Monday, at which time the senior author, Ian Lipkin, sent it to me as well.
Right.
And it's published in MBio. It is called A Strategy to Estimate Unknown Viral Diversity in Mammals. And the subtitle is Knowing the Known Unknowns. No, I'm just kidding. That was, what's his name? Rumsfeld, thank you. Anyway, there are many authors. Anthony Epstein Murray, Navarrete Macias, Zambrana Torello, Solovyov, Ojeda Flores, Arrigo, Islam, Khan, Hosseini, Bogic, Olival, Sanchez Leon, Karish, Goldstein, Luby, Morse, and that would be Stephen Morse, your buddy.
Yes.
Mazet, Dashak, Peter Dashak, right?
Very
And
well,
Ian
yes.
Lipkin.
A good man.
Big collaboration.
Wow.
So the idea here, this paper, Dickson?
I know of it. I know the cost of doing all the viruses that might occur in mammals. I've read
You
that.
probably saw that in the summary somewhere.
I did. It's a staggering amount of money.
So the idea here is to try and figure out how many viruses are out there in mammals.
Yeah, and that punchline. This is one whopper of a grant proposal.
I'll say.
Yeah.
Talk about fishing expeditions.
So the strategy was to take a single species and figure out how many viruses are in it, and then do some math, and extrapolate to all the other mammals on the
You
globe.
have to take your shoes off for that one.
What does that mean?
You got to count your toes.
Yes,
Oh.
exactly right.
Okay.
You need your toes to do the counting, in addition to your fingers.
Got it.
Everybody
That's
has to
very
take.
clever, Dickson. That's a good title. You need your toes as well? No, what did you say?
You have to take your shoes off.
Put that in the title. Show notes, please.
Sure. Sure. The ultimate fishing expedition.
Dickson, do you know what mammalian species they selected?
Bats.
Which one?
The brown bat.
No.
No. The fruit bat.
Yeah. Well, it's called
fruit bat?
This is the Indian flying fox.
Oh, okay.
Is that the fruit bat?
Yeah, that's
The Pteropus giganteus.
I think I actually saw them when I was in Australia.
Is that the way to pronounce it, Alan?
Probably.
No, you don't pronounce the P, right?
Pteropus giganteus is probably.
I have a
I would
feeling.
say it.
I have a feeling they had all these samples.
This is the one that carries Nipah virus?
Yes.
Yeah.
That was one of the reasons they say they selected it.
There is a bunch of them in Australia, I can tell you that.
But these samples were collected between 2006 and 2010. So I have a
And they
feeling.
did this in Bangladesh, right?
Yeah.
And what did they collect?
Oh, they collected
Bats.
Yeah, but
urine,
urine,
throat
okay.
swabs,
Good.
feces, and roost urine.
Got it.
Do you know what roost urine is?
The roost where they hang in the trees, but they hang upside down. How can there be urine in the trees when they hang upside down? I guess it falls down below them and hits the branches below the bats that are hanging.
So they swabbed the tree for urine?
I think they spread a tarp out below the bats.
Got it.
Yeah, that's
and
it.
sample the tarp.
There you go.
Yep. That'll work.
Got it. I hadn't gotten to the methods.
That's all right. So they have 1,897 samples.
Yeah, don't sleep under a bat-infested tree. That's all
I don't know
can
how
tell
many.
you.
I don't know how many bats that represents, but I guess they don't know.
That's an important number because because of the way the statistics are done. Multiple samples from the same bat is not going to kind of compromise things. So I think what'd you say? 1,200. How many samples?
1,800.
1,800 samples. I think it's at least 1,000 animals. It's in here. Well, I don't know if it's exactly.
So
in
they
here.
say 806 throat swabs. So that's got to be 806 different animals.
I would say at least 806 different animals.
And 926 urines, and that may overlap with the throat swabs.
So they have a huge number of samples. That's the point.
Yeah, the number of bats. I didn't find it, but it might be in here.
Right.
Yeah, their goal was to sample, and they did this over time to I assume get different seasons and all that. So they repeatedly sampled this Bangladeshi population of these bats.
Yeah.
They migrate too, don't they?
I don't know.
I think the fruit bats migrate.
Anyway, they do PCR, consensus PCR, and they only looked for viruses from 9 different families: coronas, paramyxos, hantaviruses, astroviruses, influenza A, Boca viruses, adeno, herpes, and polyoma. So they're missing some families in there.
Yeah, so they're missing, just to be specific, they're missing pox, papillomaviruses, anellovirus, if anybody cares. We don't know what they do. Both types of both DNA and RNA retroviruses. They're missing reoviruses, rhabdoviruses, filoviruses, arenoviruses, delta virus, picornavirus, calicivirus, flaviviruses, and togaviruses. So, they've got about half of the sort of standard families that affect humans Okay, so it's a sampling, but that's the nature of the beast here. They're using a sample to do to extrapolate and do an estimate.
Yeah, they wanted to sample very, very deeply in one population, in one set of viruses, and then the deeper you get that analysis, the more reliable your extrapolation gets.
By the way, was that site that they selected in Bangladesh, was that a NIPA site?
Not clear.
I don't know. Importantly, that sampling includes a large variety of viruses. So those 9 include single-stranded DNA viruses, double-stranded DNA viruses, negative-sense single-stranded RNA viruses, and positive-sense single-stranded RNA viruses. So they're getting all the major representatives.
Yeah.
So the sampling is described in a publication called the FAO Animal Production and Health Manual number 12, and I couldn't find that, so I don't know where and when. Except for the years, okay.
So tell them what FAO stands for.
Food and Agriculture Organization.
Well, it's
not FAO Schwartz.
It's not FAO Schwartz. That's exactly what it's not. They're not toying around.
So they got 985 viral sequences representing 7 of the 9 viral families. So they didn't get 2 of them. They got 11 paramyxos, including 10 new ones, and including Nipah. They got 14 adenoviruses, of which 13 were novel, 8 novel astros, 4 new coronas, 3 new polyomas. What else here? Herpes viruses. 13 distinct clades of herpes. None could be reliably classified with any genus, so they're all new. And our Boca viruses. 2 different Bocas, both of which showed greater than 98% nucleotide identity to known human Bocas.
Yeah.
It's interesting. We'll talk about that. So they use this data they apply the statistical procedures to this data to estimate, how many total viruses would be in mammals and how saturated their sampling was.
Right. And they didn't make up these methods.
Right.
These are established statistical methods that are used by ecologists and wildlife biologists to estimate species diversity. So when they go to an area and you want to say, well, how many species of birds are there in the rainforest? Well, you're not going to be able to do that whole experiment. So what you do is you do this exact sort of thing. You sample one area or one subset of species as exhaustively as you can, and then you do these statistical analyses, and they have these various algorithms, and you plot these types of curves that the more sampling you do, the fewer new things you discover.
Right.
Mm-hmm.
So the basic idea is you keep sampling until you keep getting duplicates.
Right.
And then you characterize, as you're sampling, you say, okay, how many new and how many duplicates are we getting at this time point? And then you can construct these curves, and the asymptote of the curve should be pretty close to a reasonable estimate of the total number of species there.
Yep, exactly. Thank you.
So that's what they do in non-mathematical terms.
So they estimate that there are 58 viruses in this species of flying bats, flying foxes, right?
Yep.
And they found 55 of them, so they're close to the number. Yep. And they say remaining viruses in this species are probably rare, and this is a minimum estimate. I say that's reasonable.
Yeah, absolutely. Estimate. I would say it's a minimum estimate. I'm just looking for a number here. What did we say? They had 1,800 samples.
Right.
276 contained virus. Mm-hmm. Okay, so it's that to me was interesting. It's a minority of these things that contain virus. As we have already said, they're not. This is all PCR with primers that are designed they're consensus primers, so they're designed to, based on what we know about viruses, to pick up viruses in a given family that are all related. But it's not like deep sequencing or something that's going to pick up everything. So I would say it's absolutely a minimum estimate.
Yeah, and they say that if we did deep sequencing, we'd certainly pick up more. The question is how much more? Is this a 50%? Is it, twofold or threefold or fourfold, tenfold? Who knows, right?
Yeah, so I have another question then. Is there an experimental design flaw in the selection of samples?
I don't know.
I don't see
that is
one.
To say, is there a chance for cross-contamination from other animal sources which would confound your results?
I suppose if you're looking in feces, then you could have sequences from stuff they ingested.
Exactly.
Okay, that kind of thing.
Or something that walks across the tarp that you've put down to collect the urine and urinates on it as it goes through.
Well, you could genotype the host to make sure it's just the one species, right? And throw out by mitochondrial DNA or something like that, right? I don't know if they did that.
I just can't imagine. I mean, this is 12,000 PCR reactions.
Right.
They don't mention anything about a robot here. They do have a lot of authors.
Yeah.
Okay. I mean, it's just an enormous amount of work. And when you think that you cannot afford to have any contamination with this, Vincent, have you ever been over there and looked around the lab?
Mm-hmm.
I mean, they must go to extraordinary measures to keep down contamination.
Well, they have rooms where you just do PCR and nothing else, right? Sample
they must have some
preparation.
automation too. Right.
There's some robots, but a lot of the people say the robots aren't very good. They'd rather do it by hand.
Right.
So one thing that they don't consider, and this came up last time, or maybe it was 2 weeks ago, having a PCR product does not actually mean that the virus is replicating there.
Right.
So I don't know what fraction of the positives are that, right?
Right.
And at some point, the assumption is made that every PCR product means the virus is there, but I don't think that's necessarily true, and I don't know what the rate is, 1% or 10% or what.
Well, but in this case, it's actually good to err on the side of identifying too many viruses.
Yeah, I think
because
it's
you're probably, as they say, this is we're coming up with an estimate and we're trying to construct this asymptote to a curve and then figure out what it would cost for to do this for everybody else.
I think it's fine for this, but obviously, if you have a specific virus in mind and you want to find the reservoir, then it's a different story. Yeah. So anyway, with the numbers, then they can make some interesting discussions. They say, for example, that to get 85% of this total virus population of 58, you would only need, what is this? 85% of the total richness would be achieved if another 500 samples were screened, but that another 5,500 would be required to find the remaining 15%.
Right. Diminishing returns.
Returns.
And then they say that the rarest viruses are going to be hard to find. You're going to have to sample an awful lot. And then they say, well, maybe they're not so important because if they're rare, they're not likely to be public health issues, right? So it's an interesting trade-off. It's going to just cost too much. Let's see. So then they say, okay, we assume that there are 5,486 mammalian species that are known. And if they each have 58 viruses in the 9 families, then we got 320,000 mammalian viruses awaiting discovery.
Right.
I'm not sure that's a straightforward calculation, though.
How many in humans? We must know the answer.
A lot. We have more than 58, don't we?
I'm asking the question.
Yeah.
I have no answer.
But aren't there going to be species with more or less?
Sure. Depends on where they live and how restricted
Well, that's what
their
I was
enzyme-
wondering about using
their environments
bats as.
Yeah, I would say there are more in bats than most others, right?
Because they migrate. I'm sure these bats migrate, not long distances, but they migrate from one ecotone and ecozone to another, and that would allow them the ability to pick up viruses that may be an unmigrating animal species, like, say, for instance, a tapir or a panda bear that's really restricted in its habitat and may only have 20 or something even less than that.
Dickson, are there more mammals to be discovered, do you think?
Yeah, there are. In fact, they just discovered a brand new monkey, and it's absolutely a primate. I should say it's a tiny little thing. In South America, they've actually uncovered
4
one.
inches, you're indicating.
Yeah, just about. It's a tiny, tiny little guy.
But I don't know, why say each mammalian species would have 58 viruses?
That's right. Why say that? I agree with you. I think that's
Well,
not.
the idea here was to get some kind of a boundary on this, and if you really did it up right, what you would do or started to embark on this, you'd want to sample more species of mammals.
Right.
You'd want to sample them maybe across more viral families.
Right.
But you start to get to the point where you have to say, well, how many is enough? And as you got into this, you would develop better and better estimates, kind of like that asymptotic curve that they develop in this paper. You would get closer and closer to what the real figures are. But I think what they're trying to do here is ballpark it.
Right.
And to say, because before this paper, if we sat here on TWiV and said, well, how many viruses are out there in just in mammals?
Yeah.
And how much would it take? How much work would it take? What would it cost to discover all of them? I don't know.
We would have no answer at all.
We have no answer at all. So they've gone from I don't know.
Well,
to,
you could make a guess.
Isn't our best model humans?
Well, back in 1993, Steve Morse made an estimate. He said, let's say there are 50,000 vertebrates on Earth, all right, and they have 20 viruses each, which is not far off from the 58 in the bat here, right? That gives you 1 million vertebrate viruses. That's not bad for not costing anything.
But there are some viruses that infect lots of different animals.
Yes, of course.
So that's shared,
right? So they made assumptions here that actually would probably probably inflate their final cost. They assumed 100% host specificity. And some other stuff. And if you were to actually embark on this project, you would certainly find that there are viruses that infect multiple hosts, and you're rediscovering the same things, and not everything has the same number of viruses in it.
Right.
And you might you'd probably also achieve some economies of scale that they don't account for here. You wouldn't have people hand pipetting 12,000 samples for each species.
No.
So this study cost $1.2 million, and so they say to discover, most of the mammalian viruses would be $6.3 billion.
Exactly.
Right.
Now, here's the thing that kind of is bothersome to me. They say this is cheaper than a pandemic. So I don't know I think it would be great to know all the mammalian viruses. In fact, all the viruses of all life forms out there, because you could learn so much about evolution and origins, right?
You could.
But I'm not sure it helps you with pandemics. In fact, they themselves say at the very end of this paper, we can't prevent a pandemic. We can't prevent emergence of
very big of them to say
that. Yet they want... $6 billion is a lot, and when you don't have a lot of money, as we don't these days for science, you have to prioritize. And
but they have a fallback line on that too, which is you could get 85% of the answer for $1.4 billion.
Yes, you know what
because
I say?
Of diminishing returns.
Just write a grant, and if it's competitive, you can do it. That's the way science works. We're not going to just give you money because we'd like this done. But the other issue is, if there are the X viruses in bats and other mammals, how does that help you with a pandemic should it arise, right? So in the case of Nipah, we figured out that there were bats with the virus and they were contaminating date palm sap, so we stopped that. But
mangoes.
But it still infects people because the bats are there and they contact people
and pigs
otherwise.
It infects pigs as well.
And the same thing with MERS. You want to know what the reservoir is. Maybe you can stop transmission, but maybe you can't.
Look at the West Nile virus. It's probably one of the most promiscuous viruses on Earth.
And we know where it came from, right?
We do.
Where did it come from?
It came from Central Africa.
What species?
What do you mean, which species?
What's the reservoir?
The Culex mosquito.
And where did it get it from? So we don't know.
We don't know the original mammalian.
But it's in birds, right?
Yeah.
Okay. Maybe it went from mammals to birds like last week.
Possible, but look at what it did in this country. I mean, it infected virtually every mammal, every amphibian, every reptile, and almost the entire spectrum of genera of mosquitoes.
Yeah.
So, when you look at a virus like that, you're going to find it everywhere.
What do you think, Rich? Is this a good thing to do for pandemic preparedness?
I don't think it's going to help us.
No.
That number to me is just a way of thinking about
biodiversity.
It's a way of thinking, framing what $6 billion actually means in terms of health issues. Okay?
We tend to use nuclear carriers as the result of that one.
I
for the price of one nuclear carrier, you could fund the entire NIH research budget.
I'd love to know all the viruses out there. I think it would be great information. But I don't think you need to say this would protect us in some way.
No.
I think there are scientific reasons. I think science is getting away from basic science being important, which leads to something, versus you have to be directly applicable to human health. And I think that's unfortunate. And I think that's what the authors are doing here, because they know that's how to get money these days. But, all the great stuff we have came from fundamental stuff that is getting more and more rare to do these days.
Also true. That's because the public demands a result based on their perception of the way research is, the purpose of research, and that is to benefit their lives tomorrow. And that's not the way it works. I agree with you.
The cool thing here, the other cool thing, is that some of these viruses appear to be introduced into bats from other species, like the Boca viruses.
Right.
And there are a couple of others, too, from some other animals, which are escaping me at the moment. But I
hate it when the animals escape.
One of the issues with this paper is that there are huge figures, which limits the amount of text on each page. So you have to scroll and scroll. Spillover, here we go. 5 viruses appear to be spillovers from humans, birds, or ruminants.
Yeah, there was a human bovine combo in there somewhere.
So they wonder, they said, we have observed these bats drinking from bodies of waters that are used by people, livestock, wildlife, etc. So that may be a source of exposure. So in other words, the bats are getting infected by other species. Not surprising, I guess.
You suppose bats call that a zoonosis?
Yeah, they do.
A peoplenosis.
They do.
I'm really impressed with the number of herpes viruses here. There's a very large percentage of these samples that contain herpes viruses.
Oh, no.
Or the ones of throat swabs, of 711 throat swabs they looked at, 557 of them had some kind of herpes virus in it.
Yeah. And
That's
they were
a
all
lot
new.
of herpes.
They were all new, right?
And they're all new.
Herpes gets around.
We covered a bat study for paramyxoviruses some time ago, right?
That's
there were a slug of new paramyxoviruses, but these are all new too, right?
Yeah, these
even relative to that.
Except for the Nipah, they're all new. Yeah.
Right.
So I suppose so. That's another reason why it would be cool to do this, to discover new viruses and see their relationships and evolution and so forth. I'm not saying that we shouldn't do it. I hate to have to couch it in terms of human health all the time, because we've already said that.
Yeah.
Okay, I think that's enough on that. It's a cool paper. It's very interesting. Press has certainly picked it up and run with it.
As well they should, I think.
I think so.
I think it's a very cool paper.
And now TWiV has picked it up,
now TWiV has picked it up. So, between that coverage and the TWiV bump, I think maybe they'll get the $6.3 billion.
Yeah, there you go.
I'd be happy to help them get it. Did you get your front loader, Alan?
No, but they called, and so I had to call them back, and they're going to come by at 4:30 with my front loader, which is not obscene, by the way. I just realized how that sounded.
This paper actually was suggested by 2 listeners. Adam, who thought we would find it interesting. Adam is a first-year student, a PhD student in the University of Washington in the Fred Hutch. And Jenny, who is a RN in upstate New York, and she wrote, "Of course you know about this sequencing, but in case you don't," and she sent us a link to the story, so thanks for that. And that brings us to email.
You know, we should do an all-email show sometime.
I know, but this other stuff is...
Yeah.
Well, we're
cool?
doing a series of mostly email shows. Maybe we can make a dent in it that way.
Yeah.
Alan, would you start?
Sure.
Please.
Jen writes, "Greetings all. Love the show and TWiP. Haven't gotten around to checking out TWiM yet, as this is a fairly recent discovery for me. Forgive me if this has already been addressed, as I'm only on episode 27, but I remember reading a book called Ebola a few years back about an Ebola outbreak at the Yambuku Mission Hospital in Africa. A man came in with symptoms they thought indicated malaria and received an injection of chloroquine. This man later came down with Ebola Zaire. Many patients were given injections that day with the same needle, and this was believed to have caused the rapid spread of the disease. Undoubtedly, many of the injections were likely chloroquine. My question is, as this seemed to be a particularly nasty outbreak, not that I can think of a nice Ebola outbreak, are the chloroquine doses administered for malaria not high enough to have an effect on Ebola, or does it need to be administered intravenously for an anti-Ebola effect, possibly? Many thanks, and keep up the great work."
So let's address that before you move on.
Yeah.
So chloroquine does inhibit Ebola replication in cell culture because it interferes with the endocytic pathway. It prevents the endosomes from acidifying. But I think you would need so much to give to a person to have any effect that it probably had no effect in this case at all.
Right.
Yeah.
And it's not, as far as I know, it's not been tested. So malaria, it's used for malaria.
Of course. The moment you inject it, it gets diluted, and then the virus is probably deposited locally because it's particulate as opposed to liquid. And the next thing you know, you've got not enough, even if you had enough to begin with.
Yeah.
Yeah.
All right. Go ahead.
All right. Then Jen has a postscript. "I forgot to ask about super rabies. Is this article for real, or has National Geographic resorted to junk science as well?" And she links to a story, and this does seem a little overblown. "Evolving faster than any other new rabies virus on record, a northern Arizona rabies strain has mutated to become contagious among skunks and now foxes, experts believe."
So this is so they've sequenced, isolates from foxes and skunks, but the population of these animals is increasing, so it's not surprising that you're getting more transmission among them.
Yeah, I didn't see anything here that suggested to me that this was actually anything special in terms of a strain of rabies. This is just regular old virus evolution going on.
Yeah, there's no indication that it's being better transmitted among the animals. And then they bring up the possibility of human-to-human transmission.
Yeah.
Which is crazy because it would really have to dramatically change. So I do think it's a bit alarmist.
This article's from 2009, by the way, and we're all still here.
Good point, Alan.
Yeah, so it's a bit. I agree with the who is this, Jen? Yeah, it's a bit alarmist.
Yeah,
and it's
2009 was before TWiV was really keeping an eye on journalism and virology, right? So
that's right.
This is all.
We started out pure.
We did. Yeah, we started out with sort of a frick and frack approach. Vince was the expert, and I was the naive non-virologist, and it's remained actually that way all throughout this show.
And some journalist came along and tainted everything.
I was looking at the old episodes, Alan. You came around 9 or 10, I think, right?
I think something like that, yeah.
Yeah.
Interesting. Rich, can you take the next one?
Sure. Jacob writes, "Hi all. I'm interested to hear the opinions of the TWiV team about the new section that Nature have decided to include in their journals. It is an extended data section, which allows for authors to present additional data in the online and PDF versions as if it were a results section rather than isolated in the supplementary information." And he gives a link to that announcement. I had a look at this, and I don't know. It's just another way of looking at the supplemental data. I didn't see that it was necessarily a solution to what to me is a problem of data overload.
This is a page formatting change.
Yes, exactly.
This is not a reconfiguring of the way the science is being presented. I mean, it is still supplementary information. If you pick up the print issue of Nature, these figures are not going to be there, and all they're doing is making it easier to navigate to those figures from the paper and PDF versions, which is a good idea. I like more easily navigated papers in the online edition because I'm generally reading them in the online edition.
I guess the main thing here is that it'll be on a web page rather than a PDF.
Yeah,
right.
Which is easier because you usually have to scroll through the PDF and find
a
everything.
PDF is a kludge. It's a fundamentally print-based medium, and it's designed to be printed or viewed as a printed document. And as a result, I mean, if you try to read a PDF on any screen other than a large desktop screen, you immediately run into problems.
Alan, why can't they just have the data with the paper online? Now it's a web page, so you can't argue that there's no room or it costs more or whatever.
Well,
right?
that's fine if you're an online journal. If you're MBio, you can do that. If you're Nature, you actually have to put ink on paper.
Yeah.
And so they need to create a page layout version of it that will fit in the published book. And so they do have a problem. They are still publishing in that format, and so they do have to produce PDFs.
So they need to get with it.
Yeah, and well, it's a very tough transition. You know, if you eliminate the paper journal, then is that a good plan?
Yeah, I agree. I think it is. And
I read my papers online. I think a lot of people read the papers online, but a lot of people are still reading the paper version. And, you know, there is this argument about the archivability of digital resources where some folks worry that maybe the paper version will be the only one around in 100 years.
Hey, Earth's not even going to be around in 50 years. Well, Earth will be, but it won't be populated.
Some version of it.
Yeah, we're all going to be gone.
Was that right?
Yeah.
When did I miss that fact?
We've been talking about it all the time.
Jeez, I missed,
so what are we worried about archives if we only have 50 years left? But I do think that we need to stop with the paper. I know there are lots of people that don't have internet access, but as Steve Jobs says, just do it. Don't worry about what people want.
Yeah, that's why I've got a drawer full of cables that don't fit anything.
Tell me about that. and you know, even charging devices that don't fit the latest iPhones.
Yeah.
I really resent that part.
You are a Luddite.
No, I'm not. I use my iPhone.
The next letter is from Jen. I don't know if it's the same Jen, but she writes, "Hello, TWiV team." Is that what she wrote before? No, it was a different greeting. So I know this is probably not a huge worry, but do you know if anyone has looked at a possible link between cormorant fishing, the use of cormorants to catch fish, which are half swallowed by the birds, but prevented from going all the way down by a rope tied around the base of the bird's throat, then brought back up by the fishermen in China and Japan, and H5N1. I realize farms and poultry and live markets are probably a far greater likelihood of disease transmission, but considering some of these fish are actually shipped to different parts of China for consumption, is this a worry? I do remember you saying in at least a couple of episodes that the flu can live on days for a day on a banknote, especially if they contain mucus. Thank you for the great podcasts. But no one has tried putting a banknote in a cormorant. Sorry.
I had never heard of this cormorant fishing thing.
Oh, yeah?
Oh, yeah. No, that's
It's
good.
astonishing.
Really?
It's like
That's amazing.
it's like underwater falconry.
That's cool. It's a form of enslavement.
So I looked at this in the literature, and there is very little about cormorants and flu, but I did find a recent paper where they did a sero-survey of cormorants off of Argentina.
Really?
This is not the ones near China and Japan, unfortunately, but half of them had antibodies to influenza virus as well as other viruses.
Huh.
But as far as I know, that they haven't isolated H5N1 from cormorants, and so, you know, at the moment, there's just no information, although you never know, right? Could be a threat, but right now nobody's paying much attention to cormorants.
Right.
And P.S., I know they're a bit out of date now, but I don't think I've ever heard you pick Men Against Death or any of Bertrand Ruchet's works.
I thought I did Wasn't
I don't know.
I think he may have come up at some point.
didn't he write 11 Blue Men?
Yeah.
Well, they may be
I
a
picked that.
smidge antiquated. They're still incredibly interesting
I picked that book.
from a historical perspective, and I think that most of your listeners are smart enough to realize that science is ever evolving, and a book published 50 to 80 years ago might be
wonderful.
a bit off on some of the scientific facts.
But the investigative, like Inspector Poirot, they never fail. Eventually, they get the facts right.
Right.
Well, it doesn't matter you picked it. It's always it's okay to keep
Yeah, we
picking.
can pick it again,
because,
yeah.
as Andre Gide said, no one listens anyway.
That's probably right.
So wait a minute. What is this book? I'm looking at.
Well,
for
it's
it here.
Bertrand Ruchet.
Bertrand Ruchet wrote a bunch of essays in the New Yorker on the epidemiology of diseases, but maybe I'm confusing him with another author. 11 Blue Men is a book that I recommended a long time ago, and I thought that he was the author, but I could be wrong.
Yeah.
It
Right.
doesn't come up.
I wasn't familiar with this book. He certainly wrote 11 Blue Men.
Doesn't come up
Thank
in
you.
the TWiV search.
Thank you. So 11 Blue Men is just a collection of his stories,
Alan, you are next.
Sure.
as opposed to Blue Tube Group Men.
Blue
Blue
Man
Tube
Group. Right.
Blue Men Group.
Todd writes, I've been a TWiV fan since hearing Vincent and Dick on the Futures in Biotech podcast. I immediately downloaded all the past episodes, and I'm current with TWiV and a little behind with TWIP and TWIM. TWiV and two other science and skeptical podcasts get priority, while all others are pushed to my queue for any time I'm driving or working around the house. I look forward to each week's discussions and always listen to the end. I'm so grateful that Rich decided to stop emailing from his bicycle and pull up a co-host chair. The additions of Alan, Rich, and Kathy have done nothing but fortify my appreciation of the podcast while topping off the Twix brain trust. Trust is an essential term here, and I'm sure I speak for thousands when I tell you that I thank you for your diligence and time, and the time you donate to our knowledge and good health. Over time, picks have become one of my favorite parts of the shows, and as weather geek, I pay special attention to the meteorological stuff. Alan's wind map pick is useful as well as mesmerizing as a daily destination for me, as well as two others. And he points to weatherspark.com. Says it's a customizable weather graph showing past, present, and future values. For me, becoming familiar with this site gives me a weather at a glance, so I no longer need to scroll the pages of hourly forecasts on more popular weather sites. In my browser, at least, I can spin the mouse scroll wheel to expand or contract the graph, and then drag the graph along to find a particular date and time to focus on. I tend close to the side with it.
To close the site.
I tend close to the side with the radar, so I can get more data and use my second pick for radar. Then that site is imap.tv. This is a zoomable satellite map with an array of layers on the right that you can add or subtract to let you find the type of information you're interested in. It also has icons for many storm chasers that are live streaming as they roll along. I've seen the IMAP logo flash on CNN's coverage many times in the past month or two. It's currently 55 Fahrenheit here in northern Colorado at 8 a.m. on Sunday. Several days of temperatures getting into the high 90s and possibly hitting 100 Fahrenheit are on the way.
There you go.
Cheers, Todd.
So look out for West Nile virus.
The first one of these, the beautiful weather graph site, is an amazing panel with lots and lots of data.
Neat.
And it looks like I'm going to get wet riding home.
Oh.
Wow, this is gorgeous.
Yeah, it's
Look at all
amazing.
this stuff.
I'm going to go through
Yeah,
this.
it's a lot of data.
Wow. Oh, it's
Neat.
very nice. It's very nicely done. Cool. You can go back to the 50s for history.
Oh, the archives are...
Archives, all the way back to the 1950s.
Oh, that's
I could look
great.
at the day I was born.
Really?
All right.
Anywhere?
Yeah, you just put in a location. Nice. Thank you, Todd.
I'll be darned.
This is cool.
Rich Condit.
Okay, I'm sorry. I'm a
TWiV,
little distracted
Kevin
here
writes.
with this. Yes, Kevin writes, "Hi, Twivsters. I'm a graduate student studying innate immune recognition at Harvard. Do they have innate immune recognition at Harvard? And I wanted to chime in on your discussion from TWiV 236. I think you guys may have overlooked the most parsimonious explanation. I will preface by saying that we don't fully understand what it is about nucleic acids that make them good ligands for innate immune receptors. Hang on. TWiV 236. What was that about?
That was the one where Eckhard Wimmer made a vaccine against flu by changing the codon optimization.
Yes.
Okay. I will preface by saying we don't fully understand what it is about nucleic acids that make them good ligands for innate immune receptors, so it is entirely possible that some difference in secondary structure of RNA is leading to the difference in a response. It is also entirely plausible that selective pressure would push the influenza genome to be as unrecognizable as possible. However, the way we usually think about innate recognition is that it's directed against non-specific patterns, things like double-stranded RNA or RNAs with uncapped 5' triphosphate. Our innate recognition would rapidly be rendered useless if viruses could just use different codons and therefore different secondary structured genomes for the same proteins. So I wonder if the best explanation is just that the virus is just slower to kill the host. Wild-type PR8 likely generates a robust adaptive memory response, but the mice die before the memory response is useful. Since the de-optimized viruses have all the same proteins and are presumably equally able to infect cells, they may generate an equivalent immune response to wild-type without being as lethal. A related possibility is that an infected cell is making the equivalent number of viral genomes in a cell but less able to release those genomes due to a lack of HA and NA. The cytoplasmic RNA sensors don't just detect incoming genomes, they can detect replication intermediates as well. Maybe it has nothing to do with the structure of the RNA, but rather the amount of viral RNA per infected cell. Kevin. And he says, P.S. There may be an indirect way to test if it is indeed the secondary structure of the RNA. If the secondary structure of RNA was important, presumably the wild-type influenza would have been selected for the least visible. If you mutated the codons without selecting for low availability tRNAs, you should see an increase in immune recognition without necessarily slowing the production of protein. Okay, so basically he's got two explanations for how that codon changing works. One is possibly just it changes the structure of the RNA. And it's an interesting idea that in the process of evolving to evade innate immunity that is targeting on nucleic acids, that viruses actually evolve structures that are less visible to the innate immune response. Yeah. And this may compromise that. I like that idea. And I also like the idea is that it just the viruses replicate just a little bit slower, and that's enough to so that the immune response can get ahead of the game. And I think we underestimate some of these subtleties in replication. A small effect on the ability of a virus to replicate, I think, can have a huge effect down the road on fitness.
Yes.
Yeah.
But remember, the other aspect of that paper is that this was a broadly protective response, which also requires a different explanation probably than these. But hopefully they'll be followed up and people will sort it out.
Yeah.
Hannah writes, Dear TWiV, I know you try to avoid getting stuck in an influenza rut, but you should consider doing this paper anyway because it's quite neat. That would be a good title, Getting Stuck in a Flu Rut. We have to save that for titles.
Yeah.
This is a very cool paper, I think, which I had missed. It's called Self-Assembling Influenza Nanoparticle Vaccines Elicit Broadly Neutralizing H1N1 Antibodies from the laboratory of Gary Nabel. And they basically fuse the influenza HA to ferritin, which normally forms nanoparticles composed of 24 identical polypeptides. They make these really cool structures. They're spherical. They're sort of virus-like. And so now you have a repeating HA. They immunize mice with this, and you get good neutralizing antibody responses. It's protective and broadly neutralizing against a lot of H1N1 strains. And it seems to be different in its broadly protective aspect from these stalk vaccines that we have talked about before. So I think this is pretty cool.
Yeah.
And I'm sure this is going to go further and possibly into a clinical trial because, our current vaccine isn't great and we do need something better.
So, what are the available methods for using influenza protein subunits for, rather than whole virus, either live or killed?
Mm-hmm.
You can make
Right.
the HA in plants now, right?
Right. Then you
Yep.
get virus-like particles.
Virus-like particles.
And that's in clinical trial. You can make the HA in baculovirus as just a purified protein, and that's licensed. That's
Okay.
I think that's called flu block, so you can get
Okay.
that already. How else could you do it? I think those are the ways that are going forward.
Okay, so this is
would be a
another
third way.
a third way of accomplishing the
Yeah.
same thing, and it would be terrific to have a vaccine that was actually a fairly straightforward to produce subunit vaccine, because that's going to that's going to cut the time lag
Yeah.
from identification
Yeah.
of the strain to production of a vaccine way down, because you don't have to do any of the virology.
Right.
So they took the HA from a 1999 strain and made these nanoparticles, and it protects from challenge in ferrets from strains ranging from 1934 to 2007.
So what's the basis of that broad protection? Do they
It's
say?
different from the stalk. It's different.
Okay.
They don't identify.
So it must be hitting some sort of conserved epitope.
Yes,
Yeah.
but it's not the same as the stalk. So
Yeah,
you can
something-
imagine.
I guess something about the way these nanoparticles are holding
Yeah.
the HA is
Presenting it.
exposing some epitope.
So you can imagine making an H1, an H2, and an H3, and even an H5 nanoparticle vaccine.
Yeah.
Just throw it in there and you protect it. This is
Well, I'll
very
tell you,
exciting.
The EM pictures of these things, if you injected, they're really cool. If you injected me with those things, my immune system would get really fired up.
Yeah.
They're really cool looking.
Plus, you can recycle the ferritin.
Yes.
All right.
It's good. It's a good idea. Good paper. Thank you.
I like
Hannah.
that. Yeah.
Alan.
Nicole writes, Dear TWiV team, I very much enjoy your podcast, and like most others, I'm currently working my way through the archives. When I saw the much-hyped news story about the possible discovery of woolly mammoth blood, I was listening to TWiV 100, where the Time Tree app was Alan's pick, and your discussion of how viruses are not included in this tree of life. I was wondering if this discovery actually was blood and some was preserved. It is hard to imagine any DNA or RNA remains. How would you use this sample to fill in knowledge gaps in viral evolution? What could you do with a limited amount of a well-preserved sample?
What I could do.
You could send it to Ian or Eric Delwart. That's what you could do.
Yes.
And they could sequence everything in it.
Here, here.
Yeah.
And they'd probably come up with some viruses.
This is a 10,000-year-old mammoth that was found frozen,
Yep.
well preserved in Russia. So one of the things they're very excited about is that they can get a really good mammoth genome sequence. I mean, they have one, but they had to fill it in with elephant sequence. So, so this will give you more mammoth.
At least they didn't use frog.
And then, yeah, Jurassic Park. Right?
Exactly.
So many potential jokes there.
So
Yeah.
then you could look for endogenous retroviruses. And
They tried to analyze the mammoth genome, but it wouldn't fit in the computer.
Ba-ba-dum-bum. They got some pretty hairy results, though.
you could look for viruses actually in the blood. So they found a fluid in this animal. They found something that they thought was blood, a peculiar liquid found in the carcass. So you could deep sequence that, yeah, make nucleic
Sure.
acid, see if you got
Absolutely.
some viral sequences.
You'll find something there.
And
If you've got enough material to work with.
So this is 10,000 years old, and if you could get some viruses that were circulating at the time, it would be really interesting because it's hard to, I mean, except for endogenous retroviruses or other pieces of DNA, viral DNA in our genomes, it's very hard to get anything older than 1918 flu.
Right.
So this is 10,000 years old. This would be pretty
That
cool.
would be cool. Yeah.
If this mammoth happened to be infected.
You might even get a hint as to why they became extinct.
Why did they become extinct?
I have no idea.
No?
But maybe they died from an epidemic of some sort.
Yeah, the theories I've seen for that have been pretty much habitat destruction and hunting.
Right, and global climate change.
Yeah, that's what I meant by habitat destruction.
Oh, right, okay. Because we didn't do much in the way of that.
No, we weren't building cities at that time, but we were certainly hunting them.
We were.
Jing writes, Dear TWiV hosts. It is my turn, isn't it? No.
You just read that one, didn't you?
No.
I just read.
You just read it, so
I
it's
just
Rich.
read the memo.
Sorry, it's Rich's
Me?
turn. Yeah. go
Okay.
ahead, Jing.
Jing writes, Dear TWiV hosts, I am a junior postdoc joining my current lab in summer 2012. By the end of last year, I made an important discovery, and if I can work out most of the details, it will be the end of the game, according to my advisor. He also informed everyone in the lab not to talk about my project to outsiders during its infancy. Last week, a former postdoc in my lab who was fired for multiple unethical conducts blackmailed my advisor to find him a new job. Otherwise, he would disclose what he knows of my project to everyone in the field during his postdoc application. Although it is not sure if he has done so, my advisor decided to publish what we have right now into a small journal as soon as possible. I understand that the quality of a paper should not be judged by the journal it is published in, but unfortunately, as a young scientist, I do need the impact factor to advance my career. Publishing my work right now means giving up all the experimental details for other competitors to follow up quickly, so that my chance to publish a complete story to a high-end journal is much rarer. Would any decent scientist buy his information to try to repeat somebody else's unpublished work? Should I convince my advisor just to ignore the blackmailer and resume what we are working on? The blackmailer returned to Argentina, but is there something we can do to sue him or prevent him from sabotaging my work? Look forward to your suggestion. Oh, what an awful story.
I'd ignore the blackmailer.
I would too.
What do you think, Rich?
Yeah. Basically, this makes me think what I'd want to do is, ignore the blackmailer, publish this stuff, and then go find somewhere else to work.
Yes.
It sounds like it's just a horrendous environment.
Yeah, and this idea that that somehow if people find out the project you're working on, that they're automatically going to be able to scoop you, consider that you do have a head start on this. You've already done the initial experiments that everybody else would have to repeat if they're going to try and do this, particularly if they're getting the information through this person who sounds like a real piece of work. And you're presuming that competitors would actually trust what this guy says, if they bothered to check references and would pursue that work. That seems like that doesn't seem like a good reason to put a poorly structured, rushed out paper into a low tier journal.
I agree. I
think continue working as if this hadn't happened at all. Tell the guy to go, well, it's a family show, but you get the idea. You just tell this guy you're not going to
take a long walk off a short pier.
Off a short pier. Thank you very much. Yes. And tell your advisor you want to just pursue this project and let the chips fall where they do.
Yeah, I agree. I don't think this blackmailer can do anything to hurt you, and I think you should just work really hard and finish it up.
Just focus on the job.
At any time on any project, you can make up all sorts of stories about what's going to happen if somebody or other knows something about what you're doing, and that makes assumptions about how they're going to behave. It also makes assumptions about how your project is going to turn out, and usually all of those stories are wrong.
That's right.
Yeah.
So you just do the work. The first place I have trouble on this is where the advisor tells everyone in the lab not to talk about the projects to outsiders. I suppose this is a topic for discussion, but I have a problem with that. Okay, of course I'm lucky because nobody cares about what I do.
I knew you were going in that direction. That's not true at all,
The
Richard.
chances that I'm ever going to get scooped, okay, I can tell everybody about what I do, and nobody's going to go do it because nobody cares, okay? But so I'm perhaps I'm a little insensitive to this notion that you might get scooped. But it makes this whole idea of not talking to people about what you're doing makes me uncomfortable. I don't like that.
I agree. I just don't like being secretive. I think science is supposed to be about collaborating. Unfortunately, there are people that do nasty things, but I think if you ignore them and just work and focus, you'll be okay.
That's right.
And have some confidence in yourself, too.
Yeah.
Just because somebody from a big lab it might possibly find out about this and might decide to pick it up doesn't mean they're going to beat you to publication.
Right.
It's true.
You know this. You could do this as well as they could.
Dickson, what do you think?
There was a time
I'll tell you.
when if you told people what you were doing, they would honor the fact that you were doing it.
Yeah.
Sure.
You know?
Well, I spent a lot of time at Rockefeller as a postdoc. In fact, all of my time there for the 3 years. And there were indeed some laboratories that were sworn to secrecy, and the doors were locked, and the data books were locked up at the end of the day. And it turned out that this particular laboratory that everybody knew about was headed towards the Nobel Prize winning kind of work that indeed they were eventually awarded, but it was very onerous to have to deal with them because you couldn't talk about what they were doing. They wouldn't tell you what the nature of the work was, although everybody knew what was going on. And eventually when the Nobel Prizes were awarded, it turns out if you look back on that prize, a high school student could now do that work. So it wasn't that important in the long run, but at that moment it was the most accomplished thing of its time, but it was done under the cloak of secrecy, almost as though it was part of the skunk works of Lockheed. And that's really crazy stuff. I think that's not science at that point. That's something else.
But bear in mind that all of industry pretty much works this way.
No, I appreciate that.
But they approach things a little bit differently. Where if somebody did run off and give trade secrets away, you could indeed sue them.
Yep. Well,
but I don't think in terms of that last question, I doubt there's any legal recourse here.
No.
And I say just plug away at it.
Sure. No, I totally agree.
Speaking of suing, our next letter is from Nathan, who writes, "Dear Team."
It's a subpoena.
Dear Team TWiV. Yeah, it's a
You're
subpoena.
all under arrest.
Thank you for posting my daughter's virus drawing and reading my long letter about biotech patents on TWiV 233. TWiV does a great job on many levels, one of which is the fact that conventional media can't devote the time needed to discuss, explore, and understand complex scientific issues. As you probably know, on June 13th, the Supreme Court unanimously ruled that human genes are not patentable, reversing an earlier ruling from the patent-savvy judges in the Court of Appeals for the Federal Circuit. The Supreme Court adopted the U.S. Department of Justice position, not joined by the USPTO, that cDNAs were entitled to patent protection. Amongst other things, they glossed over the cognitive dissonance associated with the similarities between cDNAs and mRNAs. I'm attaching an article from the National Law Journal regarding the decision. It quotes a friend of mine, Michael Tuskin, a JD PhD at a leading IP firm, Cooley LLP. Mike was a former patent examiner with the USPTO. I'm copying Mike on this email. Both of us would gladly join you on a future TWiV episode if you would like to discuss biotechnology patents.
That's a deep subject.
Should we take
a
him
good
up
idea.
on the offer?
Yeah.
I think it'd be fun.
I think that would be
fun. That'd be fun.
Yeah, we could discuss.
Okay, so we should add them to the guest list.
Right.
To the potential guest list, right?
And where does that reside? On the wiki, which we haven't made yet, right?
Yes, it's on the wiki. Right.
That's right.
We could discuss this patenting, this cloning patenting issue, as well as more broadly, biotech patents would be interesting to hear about it.
Yeah.
Okay, thank you. Alan.
Michael writes, Dear Twivers, after 2 years of listening to your wonderful podcast, I was wondering why there's not been any discussion of Draco. I know that the popular media has blown it way out of proportion, so I want a factual examination by the TWiV team. If the hype can be believed, every virologist will need to find some other line of work. Please fill us in on the real scoop. Thanks, thanks, Michael. Yes. So, Michael, we direct you to TWiV number 146, which is called Draco's Potion.
Yes, we did discuss it.
Yeah.
Maybe you're unable to see TWiV 146 on iTunes, and that's a problem with our RSS feed that we are working on. But in the meantime, if you go to twiv.tv, you will find it there, as you will find all the other episodes. Yeah, so if you how long ago was TWiV 146? Maybe he just missed it, or maybe he's working his way through the archive. Well, on iTunes, you can only get 150 and later, I think. So,
okay.
That's maybe why he missed it.
That may be the problem.
We use FeedBurner to generate our RSS feed, and they limit the size of the feed, which is an XML file, and we can't put more than 150 episodes on it, basically. Although we'd love to give everyone every episode for free. They are preventing us. But we're working on a solution. But in the meantime, go to twiv.tv.
Mm-hmm. So Dracos were synthetic molecules that are double-stranded RNA binding domains coupled with apoptosis-inducing domains.
Right.
Mm-hmm. Right? That could therefore detect double-stranded RNAs and kill cells.
Kill the cell, basically,
right?
Yeah. So we talked about the limitations on that show, yeah. You should check it out.
I think we still got jobs.
We do. Bottom line, you do. We do. Me, I'm next, right?
Mm-hmm. May I interrupt for just one moment?
Alan,
as with Alan,
Rich is next.
Alan has a problem at 4:30, and so do I.
Yeah, you can go.
I don't want to go yet because I want to give you my pick.
You want to do your pick?
I do.
All right, why don't you give us your pick of the week?
My pick of the week is
I know what it is.
A website called theskeptic.com.
Oh, yeah?
And on that website, you will find a video of the baloney factor, how to cut through the baloney factor. And it's the most remarkable 14 questions that if you answer the wrong way, what you're saying is total BS from start to finish. And this guy is great. He's the part of the Richard Dawkins Foundation, and the whole site is founded by that, by the way. And this guy that gives this explanation is just wonderful. So I think we should put the baloney factor, how to determine the baloney factor from the Skeptics website up as a pick of the week. That's what I think.
Here it is, the baloney detection kit.
It's fantastic.
It's not theskeptic.com.
No, well,
that gives me an ad for jewelry.
Sorry. Skeptic.org?
I got theskeptic.com.
Rich got it.
Yeah.
Skeptic.com, not theskeptic.com.
Okay.
Somebody's capitalizing
All
on
right, fine. So that's my pick of the week. I looked at this 3 or 4 times, and it's amusing and fun to watch and absolutely true. So if you're wondering if something is true or not, just check out these questions and see how they're answered.
All right.
All right. Thank you, Dickson.
You're welcome. I'm sorry that I have to go, but I have to.
Bye.
You know what I have to do right now? I have to teach a television producer how to pronounce the infectious disease organisms that they're talking about in Monsters Inside Me.
Well, you get paid, Dickson, don't you?
I do.
There you go.
I'm making use of my PhD here.
Have a good weekend.
Thank you, and same to you guys.
See you, Dickson.
All right,
Right on, bye-bye.
All right, and as Dickson just mentioned, I have to see a guy about a skid steer. A couple of minutes.
Do you want to?
I should probably go ahead and do my pick.
Okay,
right?
Go ahead.
Okay. I have picked a website. I'm always geeking out on cool photography, and this fellow, Nicholas Rusens, has some really, really neat. If you've ever tried macro photography, you know how technically difficult this stuff is. And it's some of the usual stuff, ants feature prominently in it, but he's got some really cool lighting and posing and just really neat stuff. The extreme macros section, I think I linked directly to his main website, but the extreme macros section is cool.
It's cool.
These bugs are downright scary.
Beautiful.
Yeah.
Bugs
I mean,
are
really
beautiful.
scary.
Nice. Yeah, this is very cool.
Wow. Okay.
So that's my pick.
All right.
Excellent.
Thank you, Alan.
Thank you.
You're going to leave permanently, right?
Well, not permanently, but for this week, yes.
For this week. Alan Dove is at turbidplaque.com and also on Twitter. Thanks for joining us, Alan.
Always a pleasure.
Have a good weekend.
You too.
And enjoy that front loader.
I will.
Okay.
Right, take care.
Bye. Rich, can we do 2 more short emails?
Sure. Yeah.
Let's see. Where were we?
We were at Draco's Potion. We were at Michael, right?
Correct.
So my turn, right?
That's right.
Jim writes, I hear periodic reference to funding issues in TWiV, TWIM, and TWIP, so thought your listeners might be interested in this hour-long podcast on crowdfunding by several engineers from the Engineering Commons website as well as the show notes. Regards, Jim, and Jim is our friend in Virginia who writes us frequently. And yeah, it's I haven't listened to it, podcast about crowdfunding, which is, I wonder if you can crowdfund $250,000 a year for 5 years. That would be serious crowdfunding.
That is serious. This is this is for doing science research. And they give a lot of links in the show notes to different crowdfunding sources, which is cool because I would like to crowdfund TWiV. Oh, look at this. An Indiegogo campaign raised more than $700,000 to send a bus monitor on vacation. What the hell is that? So you can raise serious money here.
I guess so.
Or we could raise $100,000 because we have 10,000 fans. If they gave $10 each, there you go.
Bingo.
And that's a minimum estimate, just like that bat paper was.
Yeah. All right.
All right, Marla writes, greetings. I am preparing an introductory pharmacology course for an upper division chemistry elective, juniors and seniors. I would like to dedicate part of the course to discussing current problems, recent breakthroughs in the field. Can you recommend one or more of your podcasts that would fit the bill? Marla is a professor at the University of Alabama, Birmingham. I don't know anything about pharmacology.
Nah, neither do I.
So I don't know if she was saying whether virology would fit in or what. But so I don't know pharmacology. However, if you were thinking of giving them a virus show, recent current problems and recent breakthroughs in the field.
I think this'll do.
This episode?
No. TWiV. Bingo.
Any specific episode?
Yeah, all of them.
Okay. Let's see. I'm going to pick one recent one that had to do with recent problems. Yeah. Here's a really good one. Pandora, Pandemics, and Privacy. The Pandora virus.
Which was, yeah.
H7N9 gain of function and access to the HeLa cell genome sequence.
Yeah, as a matter of fact, that's good. That's a nice assortment of problems.
Yeah.
That's very good.
Marla, check that one out. Okay, let's do some picks, you and I, Rich Condit.
Okay.
What do you have?
Okay, so I have something I just ran across at random. And it's actually been published in a couple of places. This one is a small article from Wired on fluorescent silk. So some clever individuals, and I also stuck a link in here to the original paper, some clever individuals have cloned at least 3 different fluorescent proteins into the main protein that's silkworms use to spin silk, fibroin. And the worms spin fluorescent silk. They've got a nice little, in the Wired article here, they've got a nice little comparison of cocoons from these 3 different worms. One's green, one's sort of orange, and one's red, and how they compare to white light and what they look like under fluorescent light. And they have a couple of models with dresses spun out of the fluorescent silk under a black light. And it's at least amusing, if not, I don't know if it's commercially viable, but it's very interesting. So there you go.
Yeah, I don't know what you would do with this.
Well, you'd have to be dancing under a black light, I guess.
Yeah, right.
Though, there are some. What it reminds me of is there are some agents that are put into whiteners in laundry detergents that actually fluoresce a little bit, and there's enough UV in natural light to give a brightness to clothing when it's impregnated with this stuff. Now, I don't know if the silk actually looks any different. Actually, it doesn't look any different under normal light, of course. Well, a little bit. It's slightly colored here, but nothing like what it looks like under a black light. So at this point, this is mostly a novelty, I think. But
when we make plasmids that have GFP, we express them. We grow up the plasmids in bacteria, to use in eukaryotic cells later. But the bacteria, when you pellet them, are slightly green.
Yeah,
under visible light.
Yeah, we have the same thing with we make viruses that express GFP proteins, so the infected cell pellets are green.
It's very cool.
Yeah. So I sent this to. I think I've mentioned this before on TWiV. When I was a graduate student, there was a postdoc in the lab, Karen Sprague, who worked on silkworms and silk fibroin. So I know a fair amount about this biology. It's very interesting. So I sent this to her. She was pretty amused.
It's pretty funny. I like that.
Yeah.
And, of course, they make transgenic fish you can buy and put in your fish tank, and they'll glow.
Right.
Put a black light on top. My pick is something called Flight Radar 24. It is a live flight tracker. And if you do nothing, just go to the website and look at how many planes are in the air at any given time.
Oh.
So I've just loaded, and it automatically loads my location, which is the New York area. And this is a real-time display of planes up in the air.
My goodness.
And there are tons of them, and if you zoom in, you can click on each one. It tells you what the flight number is, where it originated, where it's going, its altitude, its speed, its current direction. Amazing.
That is amazing. I'm really glad to have this because there have been times when I've really, my goodness, it's like a bunch of flies.
Yes. It gives you click on each one, it gives you the trajectory. It shows you exactly where it is coming from and going.
Holy cow. Cool. That is holy cow, that's amazing. I remember seeing something like this. It was a similar projection, and there was an animation that showed how this map changed with time on September 11th.
Yeah, yes, right.
When they, I thought that was a very interesting emergency response to that, where they grounded all the aircraft. They told everybody, land wherever you can right away. And the map over a short period of time just cleared. It was amazing.
Yep.
There's a whole bunch of these things buzzing over New York City, Vincent.
Oh, yes, tons of them. Lots. And they move as they move around. This is real-time tracking.
This is great. I'm going to bookmark this sucker.
Let's see what we got down in. I'm going to take a look. If you zoom out, you can see the whole country, the whole U.S. is just blanketed with airplanes.
Yeah.
Except even Florida. Yeah, I see. When you move it, it blanks. But let's see, you got nothing over Gainesville right now. There's one.
Yeah, well, we have a toy airport.
There's one flying by Alachua, which is north of you, I guess.
This is really great because I love to track flights when my kids are on them and stuff.
Yeah, of course.
So you can really get. Of course, you can use a flight tracker, but this is cooler. This is really real time.
So I just clicked on one that's going to pass by Gainesville in about 10 minutes. It's coming from Boston and going to Fort Myers.
How do you figure out where it's going?
When you click on the airplane, a little panel appears on the left, which tells you all the information.
It tells you where it's going. Right, okay.
Altitude and speed. This guy's going 447 KTs. I don't know what KT. That's kilometer. Knots, right.
Nautical miles per hour.
Which is 514. Hey, what is a knot?
A knot is 1 minute of latitude.
Oh.
Okay, so it very conveniently couples with a nautical chart.
Yeah.
And of course, the aviation guys use the same sort of thing, a nautical chart. You can't use longitude because 1 minute of longitude is a different distance depending on what latitude you're at.
Yeah.
But latitude is the same no matter what longitude you're at. So 1 minute of longitude is the same no matter what.
Nice.
And it's a little, 1 knot is like 1.1 or 1.2 miles or something like that.
Yeah, cool. So I just clicked on a plane over Orlando. It's about to land from JFK.
Wow.
It's at 2,700 feet. You can watch it descend. When I saw this, I said, I could watch this for hours. This is just amazing.
Some of these planes are bigger than others.
Yeah, they have different icons depending on...
Different icons for different planes.
Yep.
Oh,
and it will tell you what kind of aircraft it is as well.
Yeah, there's a lot of data.
Yeah. There's a big guy approaching Florida. It's a Virgin on its way from London to Orlando. And it happens to be a Boeing 747.
I'm only part way into this, but there's a little button over on the left called 3D.
Oh.
It apparently gives you a cockpit view, but you got to have the Google Earth plug-in to do it.
Oh, I see.
I'm in the middle of doing that. We got hours of fun here.
You can follow the aircraft. Whoa! You can have your screen move as it moves.
Yeah, it looks like you can be looking at Google Earth from the point of the cockpit.
Yeah.
From the perspective of the cockpit. Holy cow!
It's very cool.
All right.
So there's a lot of traffic up there, as everyone probably knew already. But
where did you come up with this? This is great.
I was just on another website that I usually check out, and it was there.
Excellent.
Just stumbled on. I said, "This is a pick of the week for sure."
Absolutely, this is wonderful. I like this.
We had a couple of listener picks that we read, I believe, and I want to read one more from your buddy Niseen.
There you go.
He writes, "I was looking for something else, and I found this site. I don't know if you found it too. It's a NIH site called Profiles in Science. Celebrates 20th century leaders in research and public health. It makes the archival collections of prominent scientists, physicians, and others who advance the scientific enterprise available to the public." It's very cool. So they have lots of famous scientists like Salvador Luria, Francis Crick, Paul Berg, Harold Varmus, Saul Spiegelman. You just click on it and you see their papers.
And a bunch of correspondence.
Very interesting letters and stuff, yeah. It's really cool. If you browse on it, as Niseen writes, you will find interesting documents and correspondence between great scientists. There are a series of letters between Aaron Shatkin and Dan Nathans and others about the famous Asilomar Conference. It's very interesting to read what they were thinking when the DNA recombination technology was in the early days. This could be a pick of the week. Absolutely.
Geez, here we have pages out of Salvador Luria's lab notebook.
That's great.
Whoa.
Very cool.
Amazing. Yeah, this is quite an archive.
Yeah, it's great. Really nice.
Pages out of my notebook wouldn't be publishable. Some of my comments on my experiments are improper.
Anyway, thank you, Niseen. That's very cool. That will do it for TWiV 249, which you'll be able to find at iTunes and at twiv.tv. And don't forget to send us your questions and comments to TWiV at twiv.tv. Our next TWiV will be live streamed from Denver, and you can find the details about that at microbeworld.org/ASM live. Richard Condit is at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. Always a good time.
And our departed guests, that doesn't sound good. Dickson Despommier, of course, is at medicalecology.com, verticalfarm.com, and trichinella.org. Forgot that last one. And Alan Dove is at turbidplaque.com and also on Twitter. I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
I may have an interruption at some point. I'm getting a skid steer loader delivered. I'm renting one.
What the hell is that?
A what?
A skid steer. This one's actually
what
a
it
mini
is. I know
loader.
what
It's
that is.
it's kind of like a little. You walk behind. This one's a walk behind design. You've seen a Bobcat. Looks like a small bulldozer.
Sure.
Yeah.
Yeah. So this thing, it's made by the same company, but it's got tank tracks on it. And you walk behind it, and it's got the bucket on the front, but it's a miniaturized sort of version. I'm going to be spreading about 10 yards of topsoil on my yard this
10
weekend.
yards.
You're renting this thing.
I'm renting this, yes. This
That's
is
right.
not something to buy.
Why don't we make that the title of the episode? What's that thing called again?
Don't buy your Bobcats,
mini
rent
loader.
them.
It's just called a mini loader?
Yeah, it's just called a mini loader. There's nothing interesting about it, unfortunately.
It's not stump grinding.
It'll be fun to drive.